Ta-chan! Look at this! This machine allows you to enter someone else's brain.
That's impossible. I don't believe it.
I'll show you. Get into the machine with me, Plasma. Campbell, press 1207.
1207.
Are we in Campbell's brain?
That's right. Let's explore around. Look, this is a neuron.
I thought we would be able to see what Campbell's thinking right now.
All of that is controlled by neurons, which are cells that transmit information in your brain and around your body. They do so through electrical impulses.
How exactly?
Normally, neurons have a membrane potential of around -70 millivolts. However, when a threshold potential of around -50 millivolts is reached, an action potential is triggered.
What's an action potential?
It's a specific change in membrane potential. During an action potential, sodium ion channels open, causing depolarization, meaning the membrane potential gets closer to zero. Then, potassium ion channels open, causing repolarization, meaning the membrane potential gets further away from zero. This only occurs in one specific region of the neuron though. For the impulse to be carried along the neuron, we need to rely on local currents.
Local currents?
When sodium ions flow in, they diffuse to and depolarize neighboring areas. This means that more areas will meet the threshold and undergo action potentials.
Hey, why aren't there action potentials in this region?
Great question. Soon after channels are opened, they are temporarily inactivated in a period called the refractory period. This ensures that the action potential is propagated in only one direction.
Oh.
Let's look at the changes in membrane potential using an oscilloscope.
What?
An oscilloscope helps us see the change in membrane potential over time. Look, when a neuron is not stimulated, the membrane potential is constant at -70 millivolts. When there is an action potential, you can clearly see that the membrane potential increases and then decreases.
Cool. H2O, why are there blobs around the neuron?
This is myelination, which makes the transmission faster. It consists of adding a myelin sheath to the neuron. This is made of special cells, such as Schwann cells, which wrap around the neuron. This way, the transmission only has to occur at the gaps called nodes of Ranvier, rather than at each point along the neuron.
Wow, that was a lot of info. I think my brain is going to grow from all of this knowledge.
Glad you learned from this trip. Now let's go back.
Wait, what's up?
To go back, Campbell needs to type in the PIN. But Campbell doesn't know the PIN.
Who would design a machine like that? Wouldn't it make much more sense if whoever is inside the brain controls the machine? I'm sorry.
It's all my fault. Wouldn't it make much more sense if whoever's inside the brain controls the machine? Wouldn't it make much more sense if I told Campbell the passcode before the trip? I'm so useless. And Campbell doesn't even know that we are trapped in here.
Don't worry, H2O. I am 101% sure that we will be able to get back safely.
How do you know that?
Because I am plasma and I am invincible. I meant we are invincible. What's that?
It seems like Campbell is having a lot of neural activity. Maybe pain. Oh, we can study it to find a way out. There can be different types of pain: mechanical, thermal, and chemical. Mechanical pain is due to pressure, such as bumping into something. Thermal pain is due to heat or cold. Chemical pain is due to chemicals, like acids or spicy food. All of these can cause action potentials in neurons.
Spicy food can cause action potentials?
That's right. Spicy food or other chemicals can open ligand-gated ion channels. These are channels that open when a molecule, which we call a ligand, binds to the channel. When they do, ions move down the concentration gradient, which can cause an action potential. There are other types of channels. Voltage-gated ion channels open when there is a change in electrical charge, and mechanically gated ion channels open in response to physical force. These stimuli can be felt by sensory neurons. This impulse is then passed down the spinal cord and then to the brain, where the stimulus is perceived as pain.
Why is it bleeping again?
More pain? Or they are taking drugs.
Drugs? Why would Campbell?
In this context, a more precise term would be exogenous chemicals, which means chemicals not produced by the body. They can have various effects on synapses.
What's a synapse?
You can review neurotransmission in this video over here. An example of an exogenous chemical are neonicotinoids. These mimic the neurotransmitter acetylcholine, but unlike acetylcholine, once they bind, they stay. Acetylcholine is often used in muscles, so when neonicotinoids are present, this means that when a muscle contracts, it stays contracted.
That's scary.
Yeah, they are used in insecticides. Another example is cocaine, which has an effect on the neurotransmitter dopamine. This is a neurotransmitter associated with pleasure and mood. Normally, after dopamine is released, the dopamine reuptake transporter reuptakes it, so dopamine is removed from the synapse. However, cocaine binds to the dopamine reuptake transporter, making dopamine stay in the synapse. This makes the postsynaptic cell be activated constantly, so it can have an impact on mood.
Does it make you very happy then?
Yeah, but that also means that it is highly addictive, and a cocaine overdose can be life-threatening.
Oh.
I've got it! We can make Campbell press the buttons by controlling the brain. For that, we need to excite and inhibit some neurons.
Wait, you can inhibit neurons?
Yup. This means that instead of depolarizing the neuron, it is hyperpolarized, making it harder for the neuron to reach the threshold potential for an action potential. Let's talk about a specific example. GABA, an inhibitory neurotransmitter. When GABA binds to the neuron, chlorine ions flow into the neuron. This further decreases the membrane potential, resulting in hyperpolarization. Let's stimulate some neurons!
Hold on, wasn't this one supposed to excite the other neuron?
You're right, depolarization has occurred in the other neuron. However, sometimes one neuron is not enough for the next neuron to produce an action potential, so we need to excite multiple neurons.
Wow! Magic!
It's not magic, it's summation. Summation is when you combine multiple neurons to produce an effect in the postsynaptic neuron. Summation doesn't have to be with only excitatory inputs. Inhibitory inputs can also participate. Oh, hi H2O, hi Plasma.
Campbell, we miss you.
I'm glad we made it back. Were you guys controlling my brain or something? Yeah?
We were stimulating neurons in your brain, but it's all for a good cause.
It's crazy to think that everything I do or think about is controlled by cells. Yeah, some argue that even consciousness arises from the interaction of multiple neurons. That's insane. Some call consciousness an emergent property because it is something apparent only when the parts interact in a wider whole.
Campbell, were you not worried about us at all?
No. H2O was on the trip.
See, H2O? I told you that we're invincible.
What do you mean we? They were just talking about me.